Hwang. Today I'll be presenting a paper titled AortaDiff, which was presented at IEEE Viz 2025. I'd like to start by explaining the motivation behind this work. When performing blood flow simulations such as CFD, the quality of the computational mesh plays a decisive role. A simulation can only produce reliable results if the mesh that defines the flow domain is geometrically accurate and numerically well constructed. In other words, a high-quality mesh enables a high-quality simulation, whereas a poor mesh can easily introduce numerical errors or physically unrealistic flow patterns. The authors emphasize that many clinically important hemodynamic indices, such as pressure, velocity, and wall shear stress, are highly sensitive to vessel geometry. Even small variations in the vessel's 3D shape or branching structure can lead to significant differences in the simulated hemodynamics. Therefore, obtaining an accurate geometric representation is essential before any meaningful clinical interpretation can be made. In this study, the authors focus specifically on aorta geometry. Their goal is to generate a patient-specific 3D aorta mesh that can be used directly in CFD workflows, ensuring that downstream hemodynamic analysis is based on a reliable geometric foundation. This paper aims to generate a patient-specific CFD-ready 3D aorta surface mesh from CT or MRI, including major branches since branching strongly shapes local hemodynamics. The mesh must be accurate, watertight, with correct topology, and smooth for numerical stability. This is challenging due to large and anatomical and pathological variability, complex branch connectivity, and noisy, limited resolution imaging. So it goes beyond segmentation to producing simulation-grade geometry. Next, let's review prior work on 3D structure generation. Methods like DPM and PVD can generate 3D shapes, often as point clouds, but for multi-branch vessels like the aorta, they may fail to preserve connectivity and topology. They are also often weakly conditioned on CT or MRI, making patient-specific control harder. A common baseline is segmentation-based modeling is segment the aorta with a 2D or 3D network, then convert the mask to a mesh using Martin cubes. Examples include TotalSeg and LowGBNet. The downside is that the resulting masses are often not CFD-ready. Branches can fragment and surfaces can be rough, so extra post-processing is typically needed. Most recently, prompt-based models like SAM have been applied to medical segmentation. They can extract vessels from clicks or boxes, which is useful interactively, but the reliance on manual prompts limits full automation and reproducibility. For vascular anatomy, especially thin supraortic branches, results can also be unstable. In contrast, AortaDiff combines diffusion, image conditioning, and contour-based reconstruction to output smooth, CFD-ready surfaces. The design is driven by data efficiency instead of generating a full aorta point cloud, which typically needs a large data set. In models of compact representation, centerline and orthogonal contours that still captures multi-branch structure. This is the AortaDiff pipeline. I will walk through each step in more detail. Let's start with the centerline diffusion model or CDM. It encodes the centerline as a 3D channel 1D signal XYZ and reconstructs it with a DDPM. To stay consistent with the scan, CDM conditions on a CT or MRI feature volume from a Monai 3D VIT. At each centerline point, it looks up the corresponding features vector, concatenates it with the noise latent L small t, and uses this to generate patient-aligned centerlines. Here's how CDM is trained. They encode the ground truth centerline image Ci0 into a latent L0. Then add Gaussian noise over time steps to get L small t up to L large T. The model takes L small t and conditioning features F samples from the VIT feature volume at the centerline point locations and learns to predict the edit noise. Training uses the standard diffusion objective MSE between the true noise epsilon and the predicted noise epsilon theta. Next, they extract visual contours along the predicted centerline. For each centerline point P, they compute the tangent TP, define the orthogonal plane phi P, and resample the CT or MRI volume onto it using RP to get a cross-sectional slice SP. Each slice is then segmented with SAM-based scribble prompt using the centerline point as an automatic prompt. Finally, they apply canny edge detection and take the outer boundary as the 2D contour. After segmentation, the contour they obtain is still in the 2D coordinate system of the cross-sectional plane. Conceptually, that contour lives on a plane in 3D with a specific location and orientation. To place it back into global 3D space, they use the inverse rotation RP and the global origin GP of the slice. For each contour point I, they transform the local coordinate IC into global space using this function. Once they map the contours into 3D and arrange them along the center line, they get the overall visual shape, but it's still just a collection of curves. To convert these curves into a smooth and continuous visual surface, they feed a NURBS model. Next, they build a watertight mesh double for CFD. They model the main aorta and each supra-aortic branch using NURBS, then clean up overlapping regions and merge them into one watertight aorta mesh. With this mesh, they run CFD in open form and obtain hemodynamic quantities such as velocity, pressure, and wall shear stress. These outputs can be used to compute clinically meaningful indicators including aneurysm, rupture risk, or potential for thrombus formation. Its experiments target four goals. First, accurate centerline generation. Second, stable and precise contour extraction. Third, producing a high-quality watertight mesh that can run CFD. And finally, evaluating how close the resulting hemodynamics are to the ground truth mass in terms of wall shear stress, velocity, and pressure. They use the VMR and AVT datasets. Next, they evaluate centerline approach. Among all compared methods, each approach achieves the best overall scores, and the qualitative result also shows that its predicted centerline is the most similar to ground truth. Here, CD, LCD, and EMD quantify geometric discrepancy between the predicted and ground truth centerlines. Lower values indicate closer agreement. Here are the segmentation metrics for contour extraction. AortaDiff achieves the best score overall. The visualization compares the differences between the ground truth and each baseline method. Next, they evaluate contour extraction and the resulting contour point class compared to other segmentation-based baseline. Their method achieves consistently lower errors indicating more accurate and stable contour reconstruction. Across all datasets, the model achieves the best overall results. In the visualization on the right, the red mesh is the ground truth, and higher overlap indicates better reconstruction quality. As you can see, AortaDiff overlaps the ground truth the most, showing the closest match. In this experiment, the authors run CFT on both the ground truth masses and the AortaDiff generated masses under the same conditions. They compare velocity streamlines, pressure fields, and wall shear stress distributions. Overall, the patterns are very similar, and they describe the agreement with the ground truth as a high degree of consistency. AortaDiff is an end-to-end pipeline that makes aorta geometry generation more automated, more accurate, and more useful in clinical settings. However, for complex diseases like aorta dissection, generating a clean CFT-ready mesh is still difficult. Next, they plan to extend the method to these lesion cases using stronger structure conditioned diffusion models aiming for more reliable CFT-ready outputs. Thank you for listening.